Hello everyone, welcome to Twashta. This question is from CSIR Net December 2019 paper from section C, and it says a mixed cell population was stained with two antibodies, one specific for cell surface antigen A and the other specific for cell surface antigen B. Anti-A antibody was labeled with fluorescein and anti-B antibody was labeled with rhodamine. The cell population was then analyzed for presence of antigens by flow cytometry. Which one of the following is the correct outcome for this cell population? So we have to figure out which one of the graphs given here is the correct option. Now, this is a very simple question from section C, and such questions offer the perfect opportunity to increase your score. Many of you might be able to answer it correctly by now, and for those who couldn't, let's get to know about flow cytometry. Flow cytometry is a very popular and powerful analytical technique that can be used to detect and quantify different types of cells in a cell population. A flow cytometer utilizes a beam of laser light and a series of detectors to detect scattered light and fluorescent signals from cells. To produce this fluorescent signal, the cells are first tagged with fluorescent markers. For example, in the question, the cells were labeled with fluorescently tagged antibodies. So the cells are first hydrodynamically focused into a very narrow stream, and every time a cell crosses the laser light beam, light is scattered. There are two types of scattering: forward and side scattering. The forward scattering is due to the size of the cell. Larger the size, more the forward light scattering. One detector is placed within a few degrees of the original path of the laser beam, which picks up this forward light scattering. A second detector is placed at 90 degrees to the path of the laser light, which detects side scattered light. This side scattering is due to the intracellular complexity of the cells. So, more the side scattering, more will be the intracellular membranous structures such as endoplasmic reticulum and mitochondria in the cell. Now, all this gives information about the physical characteristics of the cell. There are separate detectors for measuring fluorescence emitted from stained cells. When cells with fluorescently tagged antibodies bound to their surface antigens pass through the laser, the fluorescent molecules get excited by the laser. After this excitation, they emit light that is recorded by detectors. An attached computer stores all the information for each cell and integrate them all and provides us with plots or graphs for analysis. This is how we get the outcome, that is the plot. The left one is for a population of leukocytes, and as you can see, forward scatter is on the x-axis and side scatter is on the y-axis. Now try to apply what we just learned right now. Remember, more the forward scatter, larger the cell size, and vice versa. Lymphocytes are the smallest of the leukocytes. So these cells here denote lymphocytes. Similarly, we can infer for side scatter as well. Now, on the right side, we have a plot of lymphocytes stained with anti-CD antibodies on the y-axis and anti-CD antibodies on the x-axis. FL1 and FL2 here denote the two different fluorescent signals from the two antibodies. 44% of these cells are giving FL2 signal and no FL1 signal. So they are CD4 positive and are in the upper left corner of the plot. Similarly, CD8 positive cells will give only FL1 signal, and they are denoted here in the lower right corner of the plot. The 1% cells denoted in the upper right corner gave both FL1 and FL2 signal. That means they are both CD4 and CD8 positive. And likewise, cells denoted in the lower left corner are negative for both CD4 and CD8. So, with this understanding, I guess attempting such a question will be a piece of cake. Anti-A antibody was labeled with fluorescein, so it would give green signal, and anti-B antibody was labeled with rhodamine, so it would give red signal. And A giving green signal, that is anti-A antibody giving green signal and anti-B antibody giving red signal, is only in this plot. So the correct answer is option 3. Thank you.